Oh, right. Nice to be nice to hear what everyone is up to. It's been a lot of really interesting talks. So this is on behalf of the PFC task force. It'll actually be at least as much about accumbens as PFC, and largely what I think Paul has set me up really nicely for is, as we have these these interesting data sets with multiple modalities and multiple organisms and conditions, how do we go from those data to what we're trying to integrate to get to these understandings of substance use disorders and HIV. So this will be about the analytical side of that. At a broad level, right, much of what SCORCH is trying to do is to understand brain regions that are involved in substance use disorders and HIV at really 4 levels. So the first of these is, we really, for many of the regions that we're talking about, don't understand the cell types at a detailed enough level to understand, say, functional subtypes of medium spiny neurons or any other brain region. And so our first task is to put together larger data sets that have been available and to make sense of how to compare them across all these different conditions. So the second goal for SCORCH is to understand what gene expression differences and epigenomic differences are associated with disease states. I know we're all doing this, but I feel like it's, coming back to these sort of core things is helpful, right? And out of that, we hope to come to some understanding and a prioritization of all the differences that are present in the different conditions, which are the most informative, the most reproducible, the most interesting, so that we can do validation experiments to follow these things up. And the hope is that this gets us to actual, say, targets for trying to intervene in addiction or in the neurocognitive effects of HIV. And then finally, of course, the goal, in addition to all of the rest of that, is to make the data available so that anyone else in the world who is interested in these questions can take a look at the data themselves and come to their own conclusions. So the role of the NEMO SCORCH Data Center, the role of what we're trying to do in integrative analysis is to try to facilitate this process at a consortium level. And I think what makes this especially challenging, but also especially exciting, is that the effects of addictive substances, the effects of HIV, are so complex across brain regions and cell types, and different models of addiction, different phases of addiction, different phases of infection with HIV. And we can look for gene expression changes in any particular condition in one person's experiment. But our opportunity is to try to use all the data together to get to a more holistic view of that, right? So if we see that that a particular cell type is responding in one animal model, it's I think helpful to know what 5 other animal models would tell us about that same cell type, and whether, and of course, is that same effect occurring in even some of the human patients that have a substance use disorder. So I think that our challenge is to try to bring together the data to make that that sort of holistic view of the data possible. Thank you to all of you who helped put together the marker paper, which is where this figure came from. That's finally out for review at Molecular Psychiatry. Hopefully we'll all get to be a little bit more public about what we're doing relatively soon. but this is a vast amount of data that we're collecting as a group. There's over 15 brain regions for which at least one of our studies is collecting data. And overall, I think we have a really nice representation of data from regions that are involved in reward and salience and habit formation, regions of the brain that are involved in negative emotional states, and regions of the brain that are involved in executive function. So all of these different aspects of addiction will, I think for the first time, be able to look at in a comprehensive way as the data come together. So I think that that's incredibly exciting. That's but also a challenge, right? Because if addiction and HIV infection are impacting all of these regions differently, I think it's going to be, there's a lot of work to do, right? But we need to know all of that. Second kind of complexity that we face with these studies is that different human cohorts, different human patients have different kinds of exposure to addictive substances. As Paul mentioned, there appear to be really substantial effects of HIV in the brain, and so the human samples that we're studying are likely to be extremely heterogeneous. So one version of that is that we're trying to pull together samples from two rather different kinds of human brain banks. We have this wonderful opportunity to work with the National NeuroAIDS Tissue Consortium, and some of the scope of the samples that are potentially available from NNTC are shown on the left. So, what's amazing about this resource is that many of these donors enrolled in NNTC studies as years or even decades before their death, and so we have very rich information about the trajectories that they took along their path of their HIV journey, as well as their exposure to opioids and other addictive substances. So we, at least in theory, know a lot about those people's particular forms of addictions, for instance. On the other hand, many of the individuals in the NNTC cohorts are not experiencing very acute substance use disorders at the time of death, and so that's relatively few died of overdoses. So that looks like a rather different cohort in terms of substance use disorders than the kinds of donor tissues that we get when we collaborate with brain banks that focus specifically on substance use disorders, such as several of the studies have collected samples from Deborah Mash at Nova Southeastern. And so, and so we have this challenge of trying to integrate across human cohorts that might not mean the same thing. So we have that challenge for integration as well. Third, trying to understand how to use animal models to inform us about the human condition is really a fundamental aspect of what SCORCH is trying to do. I've discussed already how much heterogeneity there is amongst the human samples. The opportunity in working with the primate and rodent models is that they can mimic specific aspects of opioid use or substance use and in HIV infections in reproducible ways with lower variance, often higher quality brain samples because we don't have a postmortem interval. And so it's possible to identify more specific effects in animal models. The question being, until we make an explicit comparison into humans, we don't know whether those things are really modeling human disease. So we have to put the data together in a way that allows us to test that. And that, I think, is an opportunity, because what we are all doing is applying similar methods to all these different kinds of studies. So, okay, that's a long preamble, but I think it's important to have that frame for the strategies that we're going to take for integrative analysis. We're starting out with 2 brain regions that have been selected by the largest numbers of groups within the consortium, and trying to put all of our data together there. And those regions are the prefrontal cortex, which is being studied, I believe, by 7 of the SCORCH studies, and has really important roles in executive function and emotional control and cognition, that has well-known changes in the context of substance use and HIV. And then in addition to that, there are also 7 studies that have chosen either the dorsal striatum or the nucleus accumbens, and so we're trying to put together those data as well. Of course, the striatum and these different subregions of the striatum are central nodes in the mesolimbic dopamine reward system, and Paul has introduced that very nicely. So our goal is to figure out several things about these brain regions. So the first is, one, we need to continue to collect the raw data and metadata and make sure that everything that is being generated by the consortium, or in some cases data from outside the consortium, arrive at the data center in forms that we can start our analysis. This has been an especially large journey for the metadata, for the human metadata, but I think we're now mostly there. We still have challenges for harmonization because the data are just inherently heterogeneous, but we have quite a bit of data now, and I know, and everyone in the consortium is continuing to do amazing work, generating more that we can look at. So as we have the raw data, we are pushing those data through a uniform data processing pipeline. This is a pipeline that we are borrowing essentially for the most part from the Brain Initiative Cell Atlas Network. One of the really nice things is that there's synergy between the role that NEMO plays in SCORCH and the role that NEMO plays in the Brain Initiative, and I think that means that we can leverage these very rich, well-validated pipelines and cloud computing resources at the Broad Institute in order to do large-scale processing of SCORCH data. So we're in the middle of that. That gets us from FASTQ files, so the raw things that come off of a sequencer, to large counts matrices. Then the third part of what we do is to integrate all of these. This is not necessarily a different process than the kinds of Seurat or Harmony integration that everyone does for individual data sets. But I think that we do have interesting challenges that I'll talk a bit about, and opportunities when we're doing this across species. Because our real goal at this stage of the analysis is to find the level of annotation for cell types in each brain region where we can make comparisons across humans, non-human primates, and rodents. And I'll tell you about some of the progress that we're making there. And then lastly, I think where we're all trying to get to is, as we have these uniformly processed data sets annotated to the same cell types across them, then we can start to look at what's in common in terms of gene expression differences and higher level structures like gene co-expression networks or using tools like, say, joint decomposition methods that allow us to compare data sets in interesting ways. So that's basically what we're up to. Now I'll tell you a bit about what we've done. Okay, so as I mentioned, we're starting with prefrontal cortex and nucleus accumbens. These are not SCORCH data. These are BICCN data, but I think they illustrate exactly what we're trying to get to for any brain region for which we have cross-species data. So a couple of years ago, the BICCN was at a similar stage to where SCORCH is now, which is to say, we had cross-species data from a few brain regions, and we needed to build pipelines to figure out what would be possible to build really detailed atlases for the cell types in ways that we could compare across actually a really similar range of species. And so this study, led by Trygve Bakken and Ed Lean and a gazillion other people from BICCN, focused on the primary motor cortex in humans, marmosets, and mice. And the paper reports the level at which it's possible to identify homology across the cell types in these in these different species. And I expect that this will be, we hope that this will be the same for our data. I'll show you some evidence that it's going to be. But for instance, for glutamatergic neurons, co-integration of these data across species identified 13 evolutionarily conserved subtypes of glutamatergic neurons. These largely correspond to layer-specific subtypes that we knew about, and it appears that you can get down to about one level below the well-established major subtypes, layer-specific subtypes, to get to these 13 evolutionarily conserved types of excitatory neurons in the cortex. You can also see that it's possible, we can also look at the relative proportions of these, and overall, the same subtypes are common or rare in each of the three species, but there are significant differences in the proportions across the species that probably reflect anatomical and potentially functional differences between the M1 cortex in different species. So we're already, the point really is that when we're talking about frontal cortex regions, we already have this kind of understanding in place. So we can kind of skip this step in our data and just say, well, we kind of know the level of neuronal subclasses at which we can expect to be able to integrate the data. And it appears that that's exactly the level at which it's possible to do this integration with SCORCH data. So this is actually a human-specific integration of data coming from the Kellis group, the Sestan group, and the Spudich group, so three of our human SCORCH studies. And between those, we have close to 400,000 prefrontal cortex cells so far. That number is growing rapidly. And what you can see is that there is good integration across the studies, largely. We see a similar representation of the different subclasses that we expect to see in all of these studies. And so it appears that for prefrontal cortex, we're really in a position where we can make the comparisons that we hope to get towards an understanding of the cell type specific effects of opioids in HIV. By contrast, as Paul has so nicely summarized, although we know the major, although there's been a lot of work to atlas the cell types in the rodent nucleus accumbens, and I'll show you here that some of our progress in that. We do not have yet a comparable cross-species atlas for the subtypes of neurons in the striatum. So we're in the midst of trying to do that as really, I think, one of our major goals within the integrative analysis studies. So in rat, my group, together with David Dietz and Mary Kay Lobo, has been looking at the effects of heroin abstinence, and Ben Grissom summarized a bit of this work a few hours ago. But just briefly, we end up finding one can subset the medium spiny neurons to a very, very refined level. But we believe there are something on the order of 5 major subtypes in the accumbens that we can reproducibly find, and that seems to be consistent with similar atlases that are being assembled by other groups. As Paul mentioned, these appear to have a spatial component. So, for instance, the MSN AT subtypes, atypical subtypes, appear to be more restricted toward lateral regions, whereas the canonical subtypes appear to be more toward the core and medial regions. Consistent with work that Jeremy Day has published, we see that there are 2 major subtypes of canonical DRD1 expressing MSNs. If we're thinking about this in the context of opioids, as you can see in the dot plot on the right, there are distinct populations that express the mu opioid receptor, the delta opioid receptor, or none of the above, suggesting that, as we look for the effects of opioids, it's likely that there will be functional differences in terms of how those cells are able to respond directly to opioids. So the question is, does this same atlasing hold in human? And so these are data from SCORCH data from human nucleus accumbens collected by Manolis Kellis's group. And the initial analysis, based on really sort of the only human NAC atlas, is kind of vague, right? It just sort of, it just describes MSN subtypes, but doesn't, in a sort of generic way, but doesn't provide any real annotation for how those relate to spatial orientation or anything else. But when we integrate together a rat data with the human data, what we see is that there is essentially a one-to-one mapping of all of these MSN subtypes, which I think is incredibly encouraging. So, for instance, those 2 types of D1 populations that we already know respond differently to cocaine, from Jeremy's work, we can see there are homologs in human. For these spatially resolved subtypes, again, there's we see that human and rat essentially share subtypes of MSNs with the same markers. So I think this is incredibly encouraging, and gives us the opportunity now to go in at a relatively high level of detail, for our comparative analyses of the effects of opioids. And so just as a start to that, if we put together the effects that we find for heroin self-administration in rats, and Ben summarized this experiment, so I'll just say it briefly. Self-administration here means rats that were exposed to opioids for 10 days, and self-administration here is the time point where they've had 1 day of abstinence, so essentially a time point at which they've accelerated their drug taking and drug seeking, and then 14 days later, and what we see is that those effects are very different. If we look at the effects of human opioid use disorder, and in this case, we're looking at brains from the MASH brain bank, and many of these are overdose deaths, so individuals who have had acute exposure to opioids. What we see is that there's mostly overlap between human and this early time point of self-administration. That is a significant level of enrichment, although the odds ratio is only modest, right? Like, I guess that's probably not surprising. But we do see these, at the moment, 40 genes that are shared, differentially expressed in D1 MSNs. And if we look at what those genes are, and again, this is sort of the tip of an iceberg, I think that we get from sort of a gamish of many changes that are, maybe hard to know what to do with if we look at just the human studies or just the rat studies. But if we put them together, there's this potentially more focused thing. And, some of these genes are really strikingly interesting, right? So amongst those that are downregulated consistently between the species, we see NR4A3, a transcription factor involved in activity-dependent transcription responses that's been actually quite well described in the context of addiction. So it's encouraging to see it here. And then we have some very interesting upregulated genes like contactin 6, which is a cell adhesion molecule involved in synaptic plasticity. So this is really just like the tip of an iceberg, but I think there's a lot that we can do with these data, and I'm very encouraged that the signal seems to be there. So I'll leave it there and just think. I think we have to, for this kind of talk, have to acknowledge all of the grants and all of the people that are involved in generating these data, and and all of the Zoom calls that we come together on in order to try to make sense of them. So thank you, everyone, and I'm looking forward to getting deeper into this integrative analysis. Thanks.